going through, let's see the Glargine. So now, primarily now we will focus about Glargine clinical trials and then the various studies what we have done. So let me share my screen. Give me a second. And what you could see is primarily from the Glargine side, which we will take you through about the phase studies. In fact, what we have done is phase one and then the phase three studies, what we have done. So let's just go one by one on this perspective. Just giving a glance, though we not speak in the general diabetes section, but yes, biosimilars. What are biosimilars? As per the standard definition from the European Medical Agency, a biosimilar is a biological medicinal product that contains a version of active substances with respect to the reference medicinal product in establishing the similarity of quality, biological activity, safety, and efficacy. At the same time, we should be also aware what are biobetters. Biobetters are the improvised version of the originator biologics, but they are not biosimilars. There are other terms called as biomimics or biocopies. These are the copies of the licensed biologic medicines that have not been subjected to the rigorous clinical testing or evaluated according to the biosimilar regular pathway. And generic drugs, we all know that is a small molecule, chemically synthesized drugs using a same active ingredient with respect to the strength, dosage, route of administration, and condition for the use. And as I told you, all these three, that is biobetter, biomimics or biocopies or generic drugs, they don't categorize into a biosimilars because biosimilar or have to establish the similar in character in terms of quality, the biological activity, safety, and efficacy. Now, with that, we will move into the Biocon insulin glargine, what clinical trials we have done. This particular flow chart help you out in terms of what are the studies what we have done so far. So we did not include a couple of real world evidence, but the moment if you include the real world evidence, then it becomes quite cluttered slide to have a look at. And we as we move up into the slides, so we will speak about the critical studies, and then we don't want to do this kind of duplications of talking about multiple PKPD and multiple phase three studies. We will speak about the global PKPD studies, the global Instride 1, Instride 2, and then the Instride 3 or the extension study that we will run this up. And the same has been plotted against the global map perspective, uh, starting from BKPD study from India and then moved into Japan, Malaysia, and then the phase three studies in terms of what you say the Instride 1, Instride 2, Instride 3. So the clinical trial of Biocon Biologics, what I mean is the pivotal clinical trials of Biocon Biologics is almost encompassing almost 2,000 patients, 20 plus countries, which is of almost six clinical trial. Again, I want to emphasize that this is only the phase studies, but not the real world evidence. The real world evidence is we have done multiple countries, India, Malaysia, Europe. So it actually will have a different time to speak about the real world evidence, but in the current context, we will speak about the global PKPD studies and then the phase three of InStride 1, 2, and 3. With that, the first study is the global PKPD study. The study is done in healthy volunteers. Sorry, it is done in type 1 diabetes. It's a comparative PKPD of a proposed biosimilar glargine with a reference glargine in T1 type 1 diabetes. And this got published in the Diabetes, Obesity and Metabolism in the year 2019. It has got almost 114 patients, and then it is single center, randomized, double blind, single dose, three-way crossover. It's an hyperinsulinic euglycemic clamp study. Patients received a single dose of 0.4 units per kg of glargine subcutaneously at each time when they are into the site. So this is a wonderful study design. The study is weightaged with respect to the study design. So if you really see this, the after randomization, after the screening, the patients have been randomized, and then they've been randomized into three groups. Let's track the blue color, that is the dark blue color. There with the dark blue color is nothing but in the visit two, that is the first clamp where the patient received the biosimilar insulin glargine. In the visit three, that is during the second clamp, the patient received the same patient received the Europe referenced insulin glargine, and then the same patients in the visit four, that is the third clamp, they received the US insulin glargine. So that is where the patients have been exposed into a multiple or various insulins, that is biosimilar of Biocon biosimilar insulin glargine and also the reference insulin glargine of US and Europe. They have been at various time points. And this is very interesting. And the reason to have both US and Europe reference glargine is for the reason that we did get approval with respect to US FDA or EMEA, and we segregated data accordingly onto that, and then we ran this up. So when we finish the study design and then the patients have been given, and then when we actually look into the pharmacokinetic concentration of the insulin profiles, and you could see this, there is no significant difference between the Biocon biosimilar insulin glargine, US reference insulin glargine, or the EU reference insulin glargine. The mean serum insulin concentration profile of three insulins were similar. And we also did something called as the plasma M1 profile, that is the active metabolite of the insulins. It is still has got that kind of insulinotropic action. Again, when we measured that, the mean plasma M1 profile of the three did not differ between the two groups. That's from the pharmacokinetic perspective. Now, if you see the pharmacodynamic aspect of it, that is nothing but you use this called as glucose infusion rate, and then how much amount of glucose has been infused in the due course of time. Again, the all the three curves were superimposable. That is, came out very clearly in our study, and they did not differ between the three groups. So, in that way, the phase one study, the conclusion is the bioequivalence of biosimilar insulin glargine, that is Biocons, and then the US reference glargine and the EU reference glargine demonstrated in the patients with type 1 diabetes for both PK and PD endpoints, and they were well tolerated and no safety issues emerged. That's on the phase 1 study. Now, I'm moving up to the phase 3 studies, that is INSTRIDE 1. This is a non-inferior study comparing the efficacy and safety of bifenthrin glargine with the reference glargine in T1DM patients. Now let's see this the objective and the study design. The study design, it is a multicentric open label randomized parallel group comparative phase 3 study done in 558 type 1 patients when the screening at the HbA1c is less than 9.5 percentage, and the patients, the type 1 patients aged between 18 to 65, where they were being given once a day glargine for at least 3 months. Now, if you really look at the study design, after screening, they're running for 6 weeks to stabilize the dose of the basal bolus because of type 1 diabetes, and then they're randomized into two groups, 1:1 randomization, and then the study ran out for almost 52 weeks, and then follow for the last 4 weeks. Now, with that, with the HbA1c starting from baseline to almost 52 weeks, the HbA1c did not differ between the two groups. Being a basal insulin, we also checked the fasting plasma as a secondary endpoint, and then again you find no difference between the two groups up to almost 52 weeks. That is almost a year. Now, with the concluding of that, the least square mean difference between the Viacons Glargine and the reference Glargine were similar at 24 and 52 weeks, and they did not differ statistically, H1C. And then the basal bolus, the insulin level dose remained similar or did not differ between the two groups till 52 weeks. The adverse events in terms of the rate of hypoglycemia and any other adverse events were comparable between the two groups. So that's a very, very simplistic way of representing the type 1 diabetes. Now, going to the type in stride 2, where this is again a non-inferiority study comparing the efficacy and safety of bioconjugate glargine with reference glargine in type 2 diabetes patients. Is the insight to employed the type 2 diabetes patients. With that, it's a multicentric study, open label, randomized, parallel group, comparative three phase 3 study in TDM patients already taking on oral insulins, oral anti-diabetic drugs in 520 to 560 patients where with a population of T2DM for at least 1 year, diagnosed at least 1 year before screening, and they have been stable dose of oral antidiabetic drugs for 3 months just before screening, and they've been well stabilized with glargine side. A similar study design, what you could see after 4 weeks of screening, the patients have been randomized, where the almost 277 patients received biosimilar insulin glargine for almost 24 weeks, that is almost 6 months, and then the other arm received the reference insulin glargine for almost 24 weeks and follow for the next 4 weeks. The primary endpoint is the change in HbA1c at week 24. Secondary endpoint in terms of efficacy is change in fasting plasma glucose, change in the insulin dose, and then the SMPG perspective, is any difference in the SMPG self-monitoring blood glucose. And then the secondary endpoints in terms of safety, we have the hypoglycemia, any other adverse events, and then emergency aspect. Now, as I'm trying to talk to you about this HbA1c, that is one of the primary endpoint. Starting from baseline, you literally do not see the two lines. The lines are similar, superimposable, and they did not differ between the two groups. Now, the actual glargine dose. Again, what you could see is that it's non-significant difference. What you could see from the baseline, because they've been stabilized just before the baseline, and then starting from baseline to almost 24 weeks, the insulin dose, what you could see is for all patients is 0.12 plus or minus 0.13. You could see that for all patients for the biosimilar insulin glargine and for the reference glargine is 0.12 plus or minus 0.14. So you see literally there is not much of statistically significant difference between the glargine dose. Anyway, we'll speak about the antibodies sometime later. But concluding to the phase 3 study done in type 2 population, the mean change in HbA1c from baseline to almost week 24 is is non-inferior non-inferior to the reference glargine in T2DM patients. The change in the fasting plasma glucose level up to 24 weeks was similar. The rates of hypoglycemia and other adverse events were comparable between the two groups. Now we are coming to a very interesting study, that is INSTIDE 3 study, to study the efficacy, to assess the efficacy, the insulin dose, safety, and immunogenicity in the population with type 1 diabetes mellitus, switching from insulin glargine, switched between the bioconjugate insulin glargine and then the reference insulin glargine. So to understand this study better, we should actually we should go back to the INSIDE 1 study, where we saw that the patients who are in the reference glargine for almost 1 year, that is 52 weeks. So that particular population has been considered as a baseline for this INSIDE 3 study. So that means INSIDE 1 patients in the reference glargine become the baseline population for the INSIDE 3. So we started the INSIDE 3 from this arm of patients. And 127 patients, they have been split into two groups. So the first group is called as a switching sequence. The second group is the non-switching sequence. The switching sequences here, it talks about divided into three periods, each of 12 weeks duration. The period one, there is the biosimilar insulin glargine, because initially they got the reference glargine, right? So they've been switched to biosimilar and glargine, and they stayed for almost 12 weeks. And then from when they moved from period 1 to period 2, they again switched it to reference glargine, and then from period 2 to period 3, they've been switched from reference glargine into biosimilar insulin glargine. So that's where the three switch that took place in the switching sequence. In the non-switching sequence, they whoever has got that reference glargine, they just moved for the next 36 weeks, and then the follow period for the last 4 weeks, that is up to 40 weeks. So these are the three switching took place. The switching one took place from the baseline to the period one, and switching two took place from period one to period two, and switching three took place from period two to period. So now when we opened up in terms of the change HbA1c from baseline to 36 weeks between the two groups, they did not differ, and that's where we said that switching from a reference glargine to a biosimilar insulin glargine did not have any significant difference in terms of HbA1c. The same thing holds good as a physician. Some of the so they're concerned about will that have any aspect on the safety perspective. Again, you could see that the overall incidence of hypoglycemic events observed in two groups, no severe hypoglycemia events occurred at any point in the study. That's something which is very evident came out in the INSIGHT 3 study. The immunogenicity perspective, the cross-reactive insulin antibody percent binding over the time by treatment. Again, what you could see is that whether it is patients switching from a reference glargine to biosimilar glargine, you do not see any statistical difference from baseline till almost 36 weeks. You don't see any spikes or the troughs, and it is not statistically significant between the two groups. So that's where very glad and happy to say that in the year 2021 on July 28th, the FDA first approved an interchangeable biosimilar insulin is Biocon biosimilar Biocon's Glargine. That is a landmark approval. What we have got it on those studies, whatever that I have presented so far. So now, as I told you, we did not speak much about the immunogenicity piece, but we have a combined immunogenicity perspective that is both including inside one and inside two studies. If I directly go back to the data points, whether it is a cross-reactive anti-insulin antibody in terms of what you say from baseline to week 52, and the proportion of the patient did not did not differ in the INSTIGHT 1. And when we do this in the INSTIGHT 2 early type 2 population, again, it did not differ. The only perspective is that the value is quite high in type 1, in INSTIGHT 1. That is because it engaged the type 1 patients who are more prone to have that kind of antigen antibody reaction. And that's where the titers are quite high, but otherwise in type 2 patients inside the study, the titers level were quite low. No, so just to giving you a small history and the background. So in 2009, the first approval came from a director control of India in India that came in the year 2009, and then you could see that various approval that what we have received in the due course of time. USFA 2020, 2021 in interchangeable designation, so, and 2018 is EMEA uploaded. So that's something really makes the history of Glargine of Biocons into various stringent approvals of what we have received. As I told you about it, we are the first one to receive the interchangeable designation for the biosimilar Glargine, and the studies, whatever that we had presented, that gave that kind of what you say the perspective of approval for this particular getting a designation of interchangeable designation. And these are the various facilities, manufacturing facilities, what you could witness with the fill finish technology and fully automatized. And then we have pen assembly, completely automated line what we have, and we have pen labeling machine. Again, you could see it as a capacity of 10 million. And then we have a major, what you say, the state of art facility in Malaysia, and that's something very proud to be a part of this the insulin manufacturing perspective, which is making a landmark history. And that stop here. Feel free to ask or ping in the chat if you guys have any doubts or any comments or any questions that you guys. Thank you very much.